Okay, good day everybody. And this is Histology of the Lymphoid System. Yes, this is my lecturer slide I'll be using, because I believe her slide is very explanatory. But I'll be breaking down some things for us. Well, when you hear of the lymphoid system, what do you think about? Well, for me, I think about lymphatic. Yes, lymphatic system. It's the same thing. Yes. So the lymphatic system or the lymphoid system is made up of the lymphatic vessels and the lymphatic tissues. Now, our main course now is to now study the vessels. We're not going to have a general outline of how a lymphatic vessel is supposed to be look histologically. Okay, so we have the largest lymphatic vessel. You should know that is the thoracic duct. It is the largest. It drains all part of the body except the right part of the body, upper half quadrant. Yes, right upper half quadrant. It doesn't drain it. That is your right kidney. Your right kidney, your right lung, liver. I think kidney is not among, but right lung, liver, and head and neck, and your right arm or hand. That's it. So moving down to what we have today. So you can see that the lymphatic system includes yes, and that's what I said. So this lymphatic system now. I've talked about the lymphatic vessel. I told you the largest is thoracic duct. Now you should know how the thoracic duct. The thoracic duct is a vessel. Yes, it's made up of 3 layers. We know about the 3 layers. Every vessel is made up of tunica from the inside out. Now we have tunica intima, tunica media, and tunica adventitia, the outermost layer. Okay, but now the arrangement of the smooth muscles of the thoracic duct is quite different. It's not in a circular manner. It is arranged longitudinally. So whenever you see the slide, it will be longitudinal. Unfortunately, we don't have the slide here. Okay, so that is it, and we can classify lymphoid tissue into 2. I've talked about the vessel. Yeah, so now we are talking about the tissue. I told you we have two things that make up the lymphoid system. Is the lymphatic vessels and the lymphatic tissue. So this tissue can be diffuse or they can be dense. Diffuse means they can be discrete. Yes, they are scattered. That is diffuse. Dense means they are compacted. Okay, so the diffuse lymphoid system, what are they? Where are they found? Well, you can find the diffuse lymphoid system in your trachea, your bronchi, your urinary tract, and you can also find it in the large intestine. Just know it with anything you don't want to think of it. I think of it as trachea because we have done the histology of the trachea and bronchi, and I know that we have lymphatics there and they are diffuse. Yeah, some of them because we still see a dense lymphatic system or a dense lymphatic tissue in the bronchi. But that is different. But now we are saying that diffuse, you can find it in the large intestine. If you've done the histology of the large intestine, you will see what I'm talking about in the mucosa of the large intestine, the trachea, the bronchi, and the urinary tract. So the dense lymphoid tissue consists of an aggregation of lymphocytes arranged in form of a nodule. So they are arranged in form of a nodule. Node, you know what a node is? Circular stuff. Yes. So they are compact. That's the main point. They are together. And how can you find these dense lymphoid tissues? You can find them in an encapsulated organ, or you can find them in the mucosa. Yes, in the mucosa of organs. For instance, this lymphoid tissue can find in discrete organs that are encapsulated. Example, your tonsils, your spleen. Yes, your tonsils, spleen, your thymus, your lymphatic nodes. They are all organs and encapsulated. We are coming to that. And then we can find them in what? Mucosa, epithelium lining, all those things. Yes, like I told you, an example is bronchi associated lymphatic tissue that is found mainly in the what? Mucosa of the what? Bronchi. Note it. So we have the mucosa associated lymphoid tissue. So based on location now, if it's in the bronchi, yes, you call it bronchi associated lymphatic tissue. So going to the next slide, rather than lymphocytes, of course, you have to the embryology of the lymphocytes. Lymphocytes are cells of the lymphatic system, or lymphatic tissues, I mean to say. Lymphatic tissues have cells, just like liver have hepatocytes, the kidney has its cells, the epithelial cells, epitheliocytes, all these things are cells of particular tissues or structures. So the lymph tissue has the cells known as lymphocytes, and they can be B or T lymphocytes. These lymphocytes, where do they develop from? Well, you can say that they develop from mesenchymal cells in the yolk sac. Yes, in the wall of the yolk sac, in the liver or in the spleen. And when these mesenchymal cells migrate towards the bone marrow. So from the bone marrow, they differentiate, they form the lymphocytes. These lymphocytes enter the bloodstream. And when they enter the bloodstream, T lymphocyte will not stop. It will go to the what? Thymus first to become T lymphocyte fully differentiated into T lymphocytes. Whereas when B lymphocytes is released into the bloodstream, it doesn't go to the bloodstream. It goes to the encapsulated organ where it will stay, or maybe places where it will stay. It doesn't go to the what? Thymus for further differentiation. Okay. So that is it. So majority of the lymphocytes you're finding in your body is T lymphocytes, and they are like 85% of lymphocytes you find are what? T lymphocytes. So going down, B lymphocytes produce antibody. We should know that. And T lymphocytes, they can attack what? They can attack antigens directly by secreting maybe cytokines, cytotoxic cells. They will secrete lymphokines and cytokines. That is for that. So moving down, I've talked about the layers of this, how they are arranged. So we go to the lymphatic tissue. So remember I told you that the dense lymphatic tissue can be either encapsulated or found in the mucosa of a structure. And I give you an example. In the bronchi, we have the bronchi-associated lymphatic tissue, which is dense also, very important. And now we are now talking about the encapsulated organs. Now, what is the general feature of this encapsulated organ? What you must know that if you are a lymphatic tissue, you must be encapsulated. If you are encapsulated, you have a capsule bar. If you have a capsule, this capsule form invagination into the tissue mass or into the substance of the organ, forming septa, or what we know as trabeculae. Okay, so this septa now divides this substance or tissue mass of this particular organ into lobules. This is just the general understanding about encapsulated lymphatic tissues. So if they ask you, okay, describe the histology of the lymph spleen, or describe the histology of the tonsils, or describe the histology of the thymus or the lymph nodes, you should just use this general overview to actually describe it. If you don't remember any other thing, this will get you mark. So now the difference lies, just small differences, which we will talk about in a short while now. Okay, you have to really understand that they have similar features, but there are certain differences you have to point out if you really want to impress the examiner. You just got to memorize them. So these lymph nodes, what is special about lymph nodes? Nothing. They have capsules invaginating the tissue mass, forming septa. Yes, they are bean shaped. Lymphatic nodes or lymph nodes are bean shaped. They are like beans. You have to know it. So they also have a hilum, just like your kidney. It makes sense because blood vessels need to be entering the what? The lymph nodes. So we have the microscopic features. Okay, we have two zones of these lymph nodes. Two zones. The zones are what? The outer zone, known as the capsule, and inner zone, known as the what? The outer zone, known as the cortex. Remember, the outermost zone is the capsule that encapsulates everything. But we are now in the tissue mass. So we now have what? In the tissue mass or the substance of the lymph node, we have the outer zone, known as the cortex, and an inner zone, known as what? Medulla. This outer zone, the arrangement of the outer zone of the cortex, there is the nodule is arranged like this, such a way that densely packed lymphocytes surround a pale staining area known as the germinal center. So you can see it from this light. You can see the nodules. It's labeled here. CA for capsule. Number 2 is for the germinal center that stains poorly because it lacks numerous lymphocytes. Okay. Now M is for the medulla. So I've talked about the cortex. Now the medulla. The medulla contains blood vessels. It is worthy of note that if you see the cortex, you cannot see any blood vessel there. Yes, but these rings, you can see it. Number 4 is pointing at what? Blood vessels. Okay, that is where you find medulla. You also find lymphatics, I believe lymphocytes there too, but these lymphocytes are arranged in cord-like manner, and in the spaces are the what? Vessels, blood vessels. Okay, yes. So we should know that the capsule of our lymph node is also what? Collagen, made of collagen fiber, and beneath this capsule, what we find is the subscapular sinus. Let's try to know it. So we can move on. So as I told you, it's made up of what? Cords. Yes, I've told you about this. Then we move to the functions of the lymph nodes. Lymph nodes are responsible for draining particular area. One particular group of lymph nodes I can talk about is the axillary lymph nodes. Found in your axilla, we have the central lymph nodes, the apical, the subscapular lymph nodes, and we have the what? Pectoral lymph nodes. Okay, so all these things are responsible for draining part of your what? Axilla, and also other regions. So are responsible for draining the breast. Some are responsible for draining, the lateral side of the body, and others. So they are very important. And what makes them this important? Infection in this surrounding area immediately causes what we know as lymphadenitis. This lymphadenitis is inflammation of the lymph node. This is because when they drain that particular infected area, what happens? They drain along the bacteria, the antigen, or maybe the fungi, along with whatever lymph they are draining. And when they do so, because the lymph node is highly concentrated of lymphocytes, and I told you that lymphocytes can be B lymphocytes, they produce antibodies. T lymphocytes, they fight it by producing cytotoxins that destroy immediately. So because they are filled with this particular lymphocytes, their immune reaction immediately starts, and it is this fighting that causes inflammation of the what? Lymph nodes. Carcinoma, metastasis of the what? Cancer cells. Yes. Moving of the cancer cells is through lymph nodes. You drain. So that is why during surgical operation for removal of breast due to what breast cancer, it is suggested that surrounding lymph nodes are also what removed because they might have drained 1 or 2 cancer cells, and from there, because they are cancer cells and because they have armed themselves well, they cannot be really killed. So the process, it is drained into another lymph node. Before you know it, goes to the circulation and enters another place, causing another problem. So you see, we have talked about lymphadenditis. We are talking about the clinicals now. Lymphadenditis, carcinoma, spread of cancer, malignant cancer cells. So we have another lymphatic tissue known as the spleen. The spleen also has the general feature of the lymphatic tissue. I told you, a capsule, invagination, but now there are certain differences. The capsule of the spleen is made up more of what? Elastic fibers, yes, while that of the lymphatic node or the lymph node is more of what? Collagen and small elastin. Okay, so take note of that. During fetal life, the spleen is responsible for the production of red blood cells, but when you become an adult, your spleen becomes the graveyard of red blood cell and destroys old or aged red blood cells. It also destroys incompetent red blood cells, like we see in the case of sickle cell anemia. That's a problem, very big problem. So the spleen is covered by a layer of peritoneum. Of course, is in the abdomen. If you study your abdomen, you see that the spleen is like at the tail of the pancreas, and the stomach covers that part, and there is this peritoneal layer covering them. So the spleen is covered by peritoneum, known as the serous layer, and then except the hilum, of course, where blood vessels enter. So we also see how the capsule brings forth this invaginates into the substance, forming trabecular and then lobes and everything. Same thing, this is your spleen. Yes, we'll still see everything very soon. So this is the parenchyma of the spleen, the substance of the spleen. We also have our outer and inner cell mass, but here now we don't have the cortex and medulla like we have in the lymphatic node or lymph node. We have the white pulp and the red pulp. For me, how I remember this one is that the spleen is the graveyard of red blood cells. So we have a red pulp and white pulp. Red pulp has sinusoids, blood vessels. It is not deeply stained, okay, because it has lymphocytes and they are diffuse. They are not condensely packed, okay. So that is how you identify the red pulp of the spleen. So you can see it. Number 1 should be the red pulp, diffusely and sparsely spaced lymphocytes. That is how you find it, and you see your network of sinusoids. Sinusoids are dilated capillaries. They are big. And then you also find antigen-presenting cells near the sinusoids. So we have the white pulp. The white pulp, you can see that this actually looks like the diagram of what? The lymph node. But the difference is that you can see that these guys are like everywhere now. And then this guy that is supposed to act as our cortex now has something inside it, the reddish substance, and we'll now find out that it is what? Arterial. So you cannot find arterial in the germinal center of the pore staining center of the lymph node. No, you don't find it. So this is how you can identify that this is actually what? The spleen, not a lymph node. Presence of arteriole in the white pulp. So the white pulp has this arrangement too. It has a densely packed site of lymphocytes around the nodule, and then the polystaining germinal center. And in this polystaining area, surrounded by the densely packed lymphocytes, that is where you find your arterioles. Okay, it's not the only place though, but that is, you find arterioles there. So we have clinical correlates. Like the most popular one is splenomegaly, enlargement of the spleen. It can be caused by malaria, of course. If these malaria parasites, yes, happen to, they can be protozoa. What is it called? Yeah, and so forth. So you also have all these things because these parasites have invaded your red blood cells, and they are not normal again. Your body immediately knows they are not normal. They are destroying them, and remember, the spleen is the one destroying them. So it now leads to enlargement of the spleen. Then we have other disorders like rheumatoid arthritis, cirrhosis of the liver, splenic vein obstruction, all those things. If remember, the major drainer of the spleen is also your liver. So if anything happens to the liver, nothing is draining it, it's just enlarging. Portal vein obstruction, portal vein drains them. Okay, so all of them causes this spleen to enlarge, known as splenomegaly. So we go to the next tissue known as the thymus. Thymus is a future. It's a tissue. It is an encapsulated organ too. Okay, it weighs 10 to 15 grams and can increase about 30 to 40 at puberty. Remember, this time is obliterated as one goes by. It's just surrounded by or replaced by fat. Okay, so we have right and left lobes, which is joined by fibrous tissue. The right and left lobes are made up of connective tissue capsule, just like your normal lymphatic organ encapsulated. So you see that this is the slide for a thymus is quite different. So they say that the lobes at which it is divided into is very big, around 2 millimeters. So we have your capsule invaginating into the substance, forming a large lobe. Okay, forming a large lobe. So remember, the capsule does not reach the hilum. Let's note it. Capsule does not reach the hilum. So where were we? I've told you about the large diameter of the lobe, and they are not completely separated. That invagination of the capsule is not completely separating it, because that capsule, when you invaginate, becomes separated, dividing the tissue substance mass or tissue mass into lobes, and they are not partially separated, not completely. So we have an outer dark cortex, just like what we have known before, just the shape is quite different. Like we know before, from the spleen you have an outer cortex and an inner medulla. Here you can find it here, but here also you can see something, red mass or red structure, that's a pink staining structure that is also present here. When you see this thing, you know that it's Hassel's corpuscles. There is cells of Hassel, or Hassel's cell, or Hassel's corpuscles of Hassel, anyone. But you know that the outer cell mass, cortex, which is the cortex, is deep, darkly stained, just like the spleen, and the inner medulla is stained lightly. Okay, And now, the medulla now contains what we know as a pink staining structure. Do not mistake it for arterioles. Whenever you see thymus, the medulla contains what we know as what? Hassall's corpuscles. Please. But for our dear spleen, we have our germinal layer surrounded by the dense staining. Yes, the nodule, inside the nodule, the dense staining, we have the dense staining area, that is our white pulp. And yes, inside the white pulp we have your arteriole. Please don't forget. So the cells of the thymus are called epithelial cells of the thymus are called epitheliocytes. Okay, so you say that these cells join to form what? Internal surfaces of the capsule and surfaces of the septa and surfaces of the blood vessels. It's actually epithelial cells that form the capsules, septa, and surfaces of blood vessels in the thymus. Makes sense? It is actually the epithelial cells that form the capsule, the surfaces of the blood vessels, and even the septa. Good. So there are different types of epitheliocytes or epithelial cells depending on where they cover. Epithelial cells, how I actually memorized this was that I said epithelial cells 5 covers our Hassall's corpuscles. Hassall, Hassall's, Hassall's, yeah, we take inverted comma, yeah, Hassall's corpuscle. That is for our type 5 epitheliocytes or type 5 epithelial cells. Then for type 1, because they are number 1, cover the capsule and then the septa and blood vessels, number 1. Then number 2 will now cover the outer and inner part of the cortex. Then type 2 and 3 will cover the outer and inner cells of the cortex, and yes, type 2 will cover the outer part of the cortex, type 3 will cover the inner part of the cortex, then type 4 now will cover the medulla. Yes, the upper part of the cortex and the medulla. So type 1 is covering the outermost capsule, the septa, the blood vessels. Then type 2 is now covering the outer part of the cortex. Type 3 is covering the inner part of the cortex. Type 4 is covering the medulla, whereas type 5 epithelial cells surrounds the corpuscles of Hassall's. This is for thymus. So lymphocytes are in thymus. We already know that lymphocytes are packed. See where they are packed. Cortex. Yes, forming the dark staining area. These are lymphocyte cells. So it also says that they contain a lot of macrophages. Remember, we have macrophages like our monocytes. Yes, we have monocytes, they are macrophages. We also have our white blood cells as a macrophage, granulocytes. So think our maybe neutrophils. I don't know. So we have our macrophages. So let's continue. So this is Hassall's, the staining of what? Your thymus. Look at this very well. You can see trabeculae, vagination of this capsule, then the dark staining area known as cortex. Yes, dividing into lobe, cortex, we have the medulla. Medulla, we have our Hassall's corpuscle as number 3. Number 2 is medulla, the whole cell mass. Okay, still pink. So clinical significance of thymus, it is enlarged because of the disease associated with myasthenia gravis. We know that myasthenia gravis is disease when it's an autoimmune disease where the body cells are fighting against your acetylcholine receptors. Very bad, very bad. That is it. Move forward to the last but not the least cell, the tonsils. The tonsils are a number of collections of what tissues. They are found in your oral cavity. You open your mouth, you find your tonsils there. The main tonsils, the largest tonsils, is the what? Palatine, right and left palatine tonsils. Yes. So we have other types of tonsils. We have the pharyngeal tonsils, midline collection of lymphoid tissue. Yeah, midline is pharyngeal. And then collection are the lingual tonsils. We also have the one pharyngeal opening near the auditory tubes known as tubal tonsils. But the largest remain the palatine tonsils. So what's up with tonsils? Well, tonsils, they also have this outer mass, capsule, and what have you. But then what makes them different is the stratified squamous epithelium. The epithelium is stratified squamous epithelium, continuous with that of the mouth. Yes, stratified squamous. And pharynx is also stratified squamous. Excuse me. This epithelium extends into the substance of the tonsil. So you see that it's no longer the capsule invaginating. It's now the our stratified squamous, and it forms what we know as crypts. Apart from this, I don't think there are other different. They form crypts. So you can see the diffuse lymphoid tissue. You can see the clips. You can even see these guys. Then you can see the nodules. But you can also find out that there are no pinkish stained substance inside the germinal center. Unlike the spleen when you have arterioles, there are no arterioles here, and you can see that this forms a crypt. So this is a tonsil, side of a tonsil. Okay, so that is what you need to know about the lymphoid tissue. Thank you very much, and God bless you.